Imagine being able to use CRISPR gene editing to fix almost any spot in the human genome. I mean, even places we just couldn't touch before.
Yeah,
That's the kind of future this groundbreaking research is opening up. And it's thanks to this really innovative mix of massive biology experiments and, well, machine learning.
that's exactly right. You see, the core problem with CRISPR, the main tool, Cas9, has always been this really strict requirement.
Okay.
It needs a specific little DNA tag called the PAM right next to the spot you want to edit.
Ah, the PAM sequence.
Right. And this new work, it's published in Nature, it essentially cracks the code on that limitation. It gives us a systematic way to design custom Cas9 enzymes for, well, almost any target.
Okay, so in this deep dive, we're going to get into how they actually pulled this off, what this means for researchers, maybe like you listening, and, why it's such a fundamental shift in how we think about gene editing.
Sounds good. Okay, so let's maybe unpack the challenge first, the PAM problem itself.
Yeah, let's do that.
So basically, CRISPR-Cas9, it's this gene editing system, originally from bacteria, like their immune system.
Right, molecular scissors.
Exactly. You've got the Cas9 protein, that's the scissors, and it's guided to a specific DNA spot by a guide RNA molecule.
Okay.
But here's the catch, the really crucial constraint. The most common Cas9, the one from Streptococcus pyogenes, SpCas9,
SpCas9, yeah.
it doesn't just cut wherever the guide RNA tells it to. It absolutely must see that specific sequence, the Protospacer-Adjacent Motif, the PAM, right next door to its target site. Only then does it cut.
And for that standard SpCas9, that PAM sequence is NG, right?
That's the one. N just means any DNA base.
Yeah.
A, T, C, or G. But it has to be followed by 2 Gs.
NGG.
That
Seems simple enough on the surface.
It does. But when you're talking about editing the entire human genome, which is huge.
Billions of bases.
Exactly. That NG sequence, statistically, it only shows up about once every 16 random DNA bases on average.
So only 1 in 16 spots are even potential targets.
Pretty much. And that means there are enormous stretches of DNA, these huge blind spots, where standard SpCas9 just can't go because the NG tag isn't there.
And I bet, frustratingly, a lot of important stuff falls in those blind spots.
That's the kicker. Many really important disease-causing mutations or genes that researchers desperately want to study, they often land right inside these NG-poor regions, out of reach.
So scientists knew this was a problem. What did they try before?
Well, people definitely tried to engineer Cas9. They tried to tweak the protein, change amino acids here and there, hoping to get it to recognize different PAMs.
How did that work?
It was laborious,
Yeah.
really painstaking. You'd make maybe one change, test it, see if it worked on a new PAM, then maybe try another single change.
Just one at a time. Sounds incredibly slow.
It was incredibly slow. And even when they got lucky and found a variant that recognized a new PAM, say NGA instead of NG,
Right.
it usually only opened up a few more sites. It was very piecemeal. You still had vast regions of the genome that were just stubbornly inaccessible. They needed a totally different approach, something much bigger.
Okay, so how did these researchers break out of that slow one-by-one cycle? How did they scale this up to really tackle those blind spots?
They realized the key was understanding really systematically how lots of changes in the Cas9 protein affect which PAM it likes. Not just one change, but combinations.
So mapping the relationship between protein structure and PAM preference on a massive scale.
Exactly. The old way was like trying to file one tooth on a key and see if it fit one specific lock. These researchers decided they needed to create like a giant box of potential keys, thousands of them, and then test them all very quickly against lots of different locks simultaneously.
A giant box of keys. I like that analogy.
Yeah.
So tell us about this massive collection of Cas9s they made.
Okay, so they built this incredibly large library of SpCas9 variants. We're talking over 12,000 different versions of the protein.
What the?
Yeah, and they were strategic about it. They specifically focused on changing the amino acid bits, the residues, in the part of the Cas9 protein that was already known to interact with the DNA PAM sequence.
Makes sense. Target the area most likely to have an effect.
Right. These were the hot spots where changes were most likely to actually influence which PAM the enzyme would recognize.
Okay, 12,000 variants is a huge number to work with. How on earth did they screen all of those efficiently? How did they figure out which PAMs each one preferred? You can't do that one by one.
Absolutely not. That's where they used a really clever high-throughput experimental method. It involved a bacterial selection system using E. coli.
Okay, how did that work?
So the basic idea is this. They put each of their 12,000 Cas9 variants into different batches of E. coli. Along with the Cas9, they put in a guide RNA that targeted a DNA sequence next to a non-NG PAM. So a PAM the original Cas9 couldn't cut.
A PAM they wanted the new variants to potentially recognize.
Exactly. And here's the clever bit. They also included a gene in the bacteria that, if left intact, produces a toxic protein that kills the cell.
Ah, so if the Cas9 variant can cut the DNA next to that non-NG PAM,
It breaks the toxic gene.
and the bacteria lives.
Precisely. If an engineered Cas9 variant successfully recognizes and cuts at that specific non-NG PAM site, it disrupts the toxic gene, the toxin isn't made, and the bacteria survive and grow.
But if the Cas9 variant can't recognize that PAM,
Then it doesn't cut, the toxic gene stays active, produces the toxin, and the bacteria die.
survival of the fittest enzyme. Or the enzyme that fits the PAM anyway.
Exactly. So by setting up these experiments with different non-NG PAMs next to the target site, they could basically just grow the bacteria and see which ones survived. Survival meant the Cas9 variant inside must have worked on that specific PAM.
That's brilliant. It lets you screen thousands in parallel just by seeing what grows.
It's incredibly efficient. This allowed them to characterize the PAM preferences for nearly 1,000 of their engineered enzymes in detail. That's like 2 orders of magnitude more protein-PAM relationships figured out in parallel than anyone had managed before.
100 times more data points. That's a staggering leap in experimental scale.
It really is. And the result of all this high-throughput screening wasn't just a list of enzymes. It was this massive, rich data set.
How massive?
Over 2 million individual records. Each record detailing the activity, the specificity profile of one of these engineered Cas9 variants against different PAM sequences.
Wow.
And this huge structured data set was absolutely essential. It became the fuel for the next really exciting step, bringing in the machine learning.
Okay, so they've got this mountain of experimental data protein sequences linked to the PAMs they like. Now, train the AI brain. How did that work?
Right. They took all that comprehensive data, the specific amino acid sequence of each Cas9 variant they tested, paired with the data on which PAM sequences it actually recognized in that bacterial screen.
All 2 million records.
Yep. And they used it to train a type of artificial intelligence called a neural network. They developed their own model and gave it a name.
PAMmla.
PAMmla. PAM machine learning algorithm, I guess.
Sounds about right. And what PAMmla does, essentially, is learn the really complex underlying patterns in that data.
Patterns connecting what to what?
Connecting the subtle changes in the Cas9 protein's amino acid sequence, the structure,
All right.
to its preference for specific DNA PAM sequences. It's figuring out the rules of the game, you could say.
Like learning the physics or the energy landscape that governs how the protein shape recognizes the DNA shape.
That's a great way to put it. It learns to predict. If you give it a specific Cas9 protein sequence, PAMmla can tell you which PAMs that enzyme is most likely going to bind to and cut next to.
Okay, so it learns from the 1,000 or so characterized enzymes. But the real power comes next, right? What could they do once PAMmla was trained? Yes, this is where it gets really wild. Once the model was trained and validated on their experimental data, they could unleash PAMmla computationally. They could ask it to predict the PAM preferences for enzyme sequences they hadn't physically made or tested.
How many sequences?
A mind-boggling number. We use the model to predict the PAM preferences for potentially 64 million different Cas9 variants.
64 million. That's vast.
It's a combinatorial space that's just impossible to explore experimentally. But in silico, on the computer, PAMmla could explore this vast universe of potential enzyme designs.
So the machine learning lets them search this enormous design space, way beyond what they could build in the lab. But okay, predictions on a computer are one thing. Did they actually work? Did these computationally predicted enzymes function in the real world, like in human cells?
That was the absolutely crucial next step, the validation. They didn't just stop at the predictions.
Good.
They carefully selected a range of Cas9 variants that PAMmla predicted would have interesting, novel, or potentially very useful PAM specificities, ones different from the standard NG.
And then tested them.
Exactly. They synthesized the DNA for these PAMmla-predicted enzymes, put them into human cells in culture, and tested their performance directly.
What were they looking for?
Primarily, their ability to actually cut the DNA at target sites that were next to these predicted non-standard PAMs. That's called nuclease activity. They also checked how well these new enzymes worked when used as part of base editing systems.
Base editors being the tools that change single DNA letters without the double-strand break.
Correct. So they looked at both cutting efficiency and base editing efficiency.
And what were the results? Did PAMla's predictions hold up?
They really did.
Wow.
The enzymes designed based on PAMla's predictions turned out to be remarkably effective in the human cells.
How effective?
Very effective. The summaries mention achieving high editing efficiencies. For some of the tested variants, they were getting end delay rates. That's the measure of how often the DNA gets modified at the target site, as high as 85 to 98%.
Wow, 85–98% efficiency is excellent. But the other big question with CRISPR is always specificity, right? You want it to edit the target, not random places elsewhere.
Absolutely critical, especially if you're ever thinking about therapies. Off-target edits are a major concern.
So did they check that?
They did, rigorously.
Hmm.
They used sophisticated methods like GuideSeq to map out where else in the genome these PAMlA-designed enzymes might be cutting, even at low levels.
And what did they find?
Very encouraging results, actually.
Yeah.
What they found was that these bespoke enzymes, the ones designed by PAMlA for a specific non-NG PAM, often showed reduced off-target editing compared to some of the earlier engineered Cas9s.
Like the generalist ones that tried to accept lots of PAMs.
Exactly. Variants like SPG or SPRY, which were made to be more promiscuous and recognize a broader range of PAMs, sometimes have more off-target issues. These PAMlA-designed enzymes, being tailored for a specific PAM, seem to be more precise for that particular job.
Better precision by being more specialized. Makes sense. Okay, so they've built this incredible system. Large-scale experiment feeds machine learning, which predicts new enzymes that are shown to be efficient and specific in human cells. That's already a huge advance.
It really is.
But what if I have one very specific sequence I need to edit in the genome, and it has some really awkward PAM sequence next to it, maybe one that none of the existing enzymes, even the PAMlA predicted ones they tested, works well on? Can the system help me find or design the perfect enzyme just for my target?
That is precisely the next level they reached. They developed a method building directly on PAMlA that they call in silico-directed evolution.
In silico-directed evolution, like evolution but on the computer?
Kinda, yeah. It allows a user, like you, to basically tell the system, here is the exact DNA sequence I want to target, including this specific, perhaps tricky PAM sequence next to it.
Oh.
The algorithm, powered by PAMlA, then searches through that enormous 64 million variant prediction space, or even explores new mutations computationally to find or essentially design the optimal Cas9 enzyme variant that is specifically tailored to recognize and cut efficiently right at your exact target sequence.
So it's not just predicting possibilities, it's actively searching for the best tool for a user-defined job.
Exactly. It's like having a custom enzyme design service on demand.
That sounds incredibly powerful. Did they show an example of this?
They did, and it's a really compelling one. They used this in silico-directed evolution approach to design an enzyme for a very specific and challenging therapeutic target.
What was it?
It was a mutation in the RHO gene called P23H. This single mutation causes a form of inherited blindness, retinitis pigmentosa, and it's an autosomal dominant disorder.
Okay, autosomal dominant. Remind us quickly what that means for editing.
It means you inherit one good, healthy copy of the RHO gene from one parent and one bad, mutated P23H copy from the other parent. And just having that one bad copy is enough to cause the blindness.
So any therapy has to be incredibly precise.
Exactly. You absolutely need to disable or correct only the bad P23H copy while leaving the perfectly good, healthy copy completely untouched.
And the two copies only differ by that single base pair at the mutation site.
Just one single DNA letter difference distinguishes the bad copy from the good one, right near the PAM site they needed to target. That demands incredible discrimination from the Cas9 enzyme. It has to tell the difference between two almost identical sequences.
Wow. That's a needle in a haystack problem for an enzyme. Could their system design one that specific?
Amazingly, yes. Their in silico-directed evolution process allowed them to computationally design this Cas9 variant that was highly selective. It preferentially edited the disease-causing RHO P23H allele while largely sparing the healthy allele.
And they showed this worked.
They showed it worked effectively in human cells in the lab, achieving that critical allele-specific editing. And even more impressively, they demonstrated it in vivo. They used it to successfully edit the target allele in the eyes of mice carrying the human P23H mutation.
In living animals. That really showcases the potential here for designing these truly bespoke editors for tough therapeutic targets.
Absolutely. It moves beyond just finding enzymes for general PAMs to crafting a perfect tool for a very specific, challenging genomic location and context.
This feels like It's more than just finding a few new tools for the toolbox. It feels like they've built a whole new way to make the tools.
I think that's exactly the right way to look at it. This study isn't just another paper reporting, hey, here's a new Cas9 variant. It represents a real technical disruption, a paradigm shift.
How so?
They've built a complete platform. You could call it an experiment AI feedback loop or an engineering pipeline. It fundamentally changes the process. We move away from that slow, painstaking, trial-and-error method of protein engineering.
The one-at-a-time approach.
Right. And we move towards a systematic, data-driven, computationally guided approach for creating Cas9 enzymes with desired PAM specificities.
So the real innovation is the process, the pipeline itself.
Precisely. And this pipeline is what has the potential to unlock editing across potentially over 90% of the human genome, those vast regions that were previously off limits because they lacked that standard NGPM.
Making almost the entire genome targetable. The implications seem huge.
They really are across multiple fields. Think about gene therapy. This dramatically expands the number of genetic diseases caused by mutations in those previously inaccessible regions that we could potentially target and correct.
Okay.
For basic researchers, it just offers incredible flexibility. You can now design experiments to study gene function or the effect of specific mutations almost anywhere you want in the genome.
No, we're being limited by where the NGGs happen to be.
Exactly.
Yeah.
And even thinking beyond medicine, in areas like agriculture, imagine the potential for precisely engineering multiple traits in crops by targeting specific gene combinations that might have been impossible to hit simultaneously before.
It also really highlights this powerful synergy, doesn't it, between doing the biology experiments on a massive scale to generate the data, and then using machine learning to learn from that data and explore possibilities you couldn't reach otherwise.
It's a perfect example of that synergy. This kind of collaboration, often involving multiple labs, sometimes even industry partners working together, shows how combining large-scale experimental biology with sophisticated computation can accelerate discovery at a pace far beyond traditional methods.
It's the combination that unlocks the breakthrough. Now, you mentioned potential challenges or limitations.
Yes, and the authors are appropriately cautious, which is good science. While this platform is incredibly powerful, it's not perfect yet. For instance, they noted that some of the newly designed variants, maybe ones targeting RYG PAMs, while great at their specific target, might not be quite as universally precise across all potential off-target sites as a highly optimized generalist enzyme like PRY when tested broadly in mammalian cells. So there can be trade-offs.
Specialization versus generalization.
Kind of. And of course, for any potential therapeutic use, especially with engineered proteins from bacteria, long-term studies are always needed to look at potential immunogenicity. Does the patient's immune system react negatively to this foreign protein over time? That's always a key consideration for bringing these things to the clinic.
Right, crucial safety checks down the line. But fundamentally, even with those caveats, this feels like a game changer for how we design gene editors.
I agree. It really is a technical platform disruption. It moves us beyond just making incremental improvements to existing tools and provides a whole new framework for engineering these essential molecular machines.
So there you have it, a fascinating story of how researchers combined massive experimental screening, testing thousands of protein variants, with the predictive power of machine learning to essentially decipher the operating code for CRISPR's PAM requirement.
Yeah.
They've built a system, PAMmla and the in silico evolution pipeline, that allows them, and potentially others, to design a custom-tailored Cas9 enzyme for almost any specific DNA sequence you can think of.
Opening up those vast, previously inaccessible regions of the genome for precise editing.
It really brings that vision of truly precise, fully customizable genome editing much, much closer to being a widespread reality. You can now seriously envision tailoring the editing tool exactly to the specific DNA target you need to modify.
Maximizing efficiency, hopefully minimizing those off-target risks by designing the right tool for the right job.
Which, of course, leads us to a final thought for you, our listeners, to consider. As we gain this remarkable, almost on-demand ability to precisely target and potentially edit nearly any point in the genome, what do you think are the most critical applications? Where should we be focusing this incredibly powerful new capability in medicine, in research, maybe elsewhere, to ensure it's used wisely and for the greatest benefit?